Good afternoon, students. Am I audible? Can one of you kindly confirm?
Yes, sir, you are audible.
Okay, thanks. So I welcome you to today's tutorial session on the contents of week 6 of this NPTEL course, Introduction to Cell Biology. So as we have done in the previous tutorial sessions, in this tutorial session as well, I will first try to give you a summary of the contents of this week. So, just like the previous week, this week initially started with the continuation of the cytoskeletal elements, right? So, as we had discussed in the previous tutorial session as well, that there are different cytoskeletal elements or components of the cell, which primarily are of three different types, which are the microtubules, the actin filaments, and the intermediate filaments, right? And we had also discussed what are the different functions of what are the subunits, what are the protein components of these different cytoskeletal elements. The microtubules are formed as a result of polymerization of the tubulin subunits, the alpha tubulin and beta tubulin subunits. The microtubules are hollow tubule-like structures which act as the major highways for cellular traffic, right? For transport of membrane enclosed organelles, for the spatiotemporal localization of membrane enclosed organelles, for the transport of membrane bound vesicles within the cell in a certain direction, uh, uh, for the anchoring of the membrane enclosed organelles, as well as the microtubules in the spindle fibers, for example, they act as important, they play important roles in the segregation of the sister chromatids of the chromosome during cell division. On the other hand, the microfilaments, the actin microfilaments were made up of the actin subunits. They were formed, they were dynamic structures which are formed as well as disassembled because of the assembly and disassembly of the actin subunits again in a directional manner, in a polarized manner. And then we had not discussed about intermediate filaments. We just mentioned that there is also a third category of cytoskeletal elements within the cell, which are called the intermediate filaments, which can be quite diverse in terms of the functions, the cellular, subcellular localization, as well as the components, that is, the protein components with which these intermediate filaments are made up of. So, primarily, now we mentioned that the cytoskeletal elements, as we had discussed earlier, plays several different functions. Primarily, they play an important role in the cellular mechanobiology. That means they play important roles in providing mechanical strength to the cells, mechanical strength to the plasma membrane, which is made up of primarily lipids. So it's quite wobbly structure, which needs this mechanical support from the cytoskeletal elements. Apart from that, the cytoskeletal elements play important role in the cellular organization, right? In the intracellular organization of this membrane by anchoring of these membrane-bound organelles and the transport of the organelles within the cells, as well as, for example, these actin filaments, they're necessary for even important activities of the cell, like maintaining the cell shape, as well as in the membrane bending or membrane curvature during endocytosis, or in transducing force during cell migration, or therefore important for cellular motility, as well as in maintaining inter-cell contacts, like in the cell junctions, like gap junctions, tight junctions, etc. Regarding the actin, so we had also discussed that the cytoskeletal elements, be it microtubules, be it the actin microfilaments, they are very dynamic. Dynamic in this sense that there are two opposing things that are going on simultaneously. There is polymerization of the subunits of the units of or the protein subunits, which are basically units of these cytoskeletal elements. There is polymerization as well as there is depolymerization. So that now the balance between, that is the rate of rate at which polymerization is happening versus the rate at which depolymerization is happening from the two different ends of the cytoskeletal elements governs the length of the cytoskeletal elements, be it microtubules or the microfilaments, as well as the directionality or the distribution of these elements, cytoskeletal elements within the cell. And they're regulated, this regulated dynamics, which in turn regulate the directionality as well as the distribution or the lengths of these cytoskeletal elements, is necessary for governing and regulating cellular phenotypes. For example, the actin filament, which lies just beneath the plasma membrane and plays important role in maintaining the cellular shape as well as membrane bending or membrane invagination during endocytosis, or extension of the membranes like formation of this philopodia for cellular mobility or cellular movement, or for phagocytosis, for example, for engulfing some bacteria, extracellular bacteria, which is present in the extracellular matrix or extracellular space. So there what happens is, as we mentioned, the actin microfilaments have a very unique capability of forming of forming a mesh or network of branched actin as well, right? It becomes like a kind of a hand-like structure because of this branching of actin, which forms a very branched mesh-like network. So the in short,
Sir,
what I'm trying to say is that, Shom.
what is the cofilin and the ARP complex that is
Yeah,
given in the diagram?
that's what I'm going to that's what I was going to speak about. So basically, there are several accessory proteins. Like actin is the subunit or the protein monomer. It's a unit which gets polymerized to form the actin filaments, right? Actin microfilaments. But there are several accessory regulatory proteins which regulate the polymerization and depolymerization kinetics of the actin microfilament. So cofilin is the protein. So basically, cofilin and the cofilin is a protein which binds. There are two proteins. One is cofilin and one is profilin. So these bind with the actin subunits. Okay. So when cofilin binds with the actin subunit, it promotes the polymerization of the monomeric actin subunits to form that or elongate the actin microfilaments. That is, increase their length. Whereas profilin, Profilin, profilin are the proteins which when bind the actin subunits, or they can even bind these formed at the minus end of these long microfilaments, actin microfilaments, and cause the cleavage of the microfilament to promote the disassembly. The profilin promotes disassembly, whereas cofilin promotes actin assembly, that is actin polymerization, leading to elongation of the actin fibrils in a certain direction, right? That will be necessary for example, you think that if there needs to be a membrane extension, right? For example, you think of this, that this is a cell and you have this actin microfilament network just beneath the plasma membrane. Now, if there is this bacteria outside in the extracellular matrix, which this cell now wants to engulf, so what has to happen? Its membrane has to extend like this, right? So that for that, there needs to be elongation of this actin network in this direction, right? So that if this membrane gets a mechanical without which this membrane will not be able to elongate like this for engulfing the bacteria. So there, this cofilin will promote the actin polymerization in this direction, so that there is increasing length of the actin microfilaments and eventually providing the membrane the necessary mechanical support for it to extend and form these projections for engulfing the bacteria for the phagocytosis. As well as there are this class of enzymes which are called the ARP23 as well as other proteins like WASP, which promote the branching of the actin, right? So there is this linear actin microfilament, ARP23, and along with the WASP, they promote the branching of the actin filaments. So for these cytoskeletal elements, so some are the subunits like the tubulin for microfilament for the microtubules, actin for the microfilaments. So they polymerize to form these cytoskeletal assemblies, but there are several accessory proteins which are regulatory proteins. which regulate the kinetics of assembly or disassembly as well as branching in case of actin microfilament, right? So,
Okay, sir. Sir, one more question, sir.
yeah,
In WBCS, there is also phagocytosis. Is there any cophilin or propylene
yes,
present in WBCS?
yeah, WBCs, you mean? Yeah, I mean, in case of WBC, see, these cytoskeletal elements are present in all the cells, right? The cytoskeletal elements, I mean, microtubules, microfilaments, and intermediate filaments. Intermediate filaments, the composition can be different in different cells or different tissue types. But these microtubules and microfilaments, these tubulin and actin microtubules and microfilaments are present in all the cells. Right? The regulation can be a bit different from cell to cell. And definitely they are present in these macrophages and dendritic cells, right? Which are basically white blood cells, which are part of your innate immune defense response, which are necessary for the phagocytosis of these pathogens, and even in the neutrophils as well. I mean, they're present in all the cells, but the regulation can be these accessory proteins which are involved in specific regulation of these cytoskeletal elements can be a bit different because there are also several signaling cascades, right? I mean, the cell has to first recognize, okay, that there are these bacterial cells outside or something that is outside that it needs to engulf, and then that has to trigger, right? This signaling cascade. There has to transduction of the signal of this presence of this information that there is this bacteria outside to certain cellular activities. So that then trigger this polymerization of actin and I mean complete reorganization of the cytoskeletal elements to eventually lead to this membrane ruffling and membrane extensions to eventually give rise to phagocytosis.
Okay,
So
sir.
does
Sir,
that answer the question? So basically,
so what is the
yeah, the
what is
cytoskeletal
the
elements are present in all the cells. There's a regulation, depends on several signaling cascades as well. So that can be a bit different from cell to cell.
okay, sir. Sir, what gives the signal of the WBCS for the disease causing bacteria or the that
Yeah. So for example, so basically this is, I mean, I would like to tell you a very simple theme of signal transduction. So in signal transduction, what happens, for example, if this, consider that this is your plasma membrane, right? So on the plasma membrane, there are several of these transmembrane proteins or integral proteins, right? So a class of proteins are there which are called receptor proteins, you know. These receptor proteins are actually transmembrane or integral membrane proteins. So they are such that this is the extracellular space, okay? This is the extracellular space and this is the cytosol, right? Or the cytoplasm basically. So what happens is, for example, if you have a bacteria in the extracellular space, then there are several molecules of the bacteria which are not present in the human cells, right? They are unique for the bacterial cells. For example, let's take the example of lipopolysaccharides. If it's a gram-negative bacteria, then it has lipopolysaccharides. Or if it's a gram-positive bacteria, for example, then it has teichoic acid in its cell envelope. Or several, in case of gram-negative bacteria, it can also be several outer membrane proteins, right? So there are several cellular receptors. For example, I can give you an example like toll-like receptors, right? TLR. So these receptors actually recognize these several of these bacterial proteins, pathogenic bacterial proteins, right? That bacterial proteins, or for example, this lipopolysaccharide, which are the outer membrane, that is the bacterial cell envelope components. So on recognizing that, so this is an extracellular signal, right? But when it recognizes this signal, which means that there is presence of bacteria in the extracellular space, what it does is there is some change of conformation of protein conformation in the cytosolic end. Okay. When there is binding of this ligand, there is some conformational change of this protein at the cytosolic end. And then what happens is several other proteins get recruited and activated in the cytosolic end, which eventually causes a cascade or a relay of signal, right? Basically, there are several messengers you can think of. Like this is the guard at the main gate. He has seen
means
something
it
going wrong in outside the campus or outside the house, and then what he does is he conveys the signal to the inside of the house that something is going wrong outside, And
Means it does
then
like the function of the neuron, like the signaling function of the neuron. Same function only.
yeah, I mean,
Signaling.
this signaling function is, the signaling function exists in almost all the cells. Just the ligands are different, the cellular responses are different. For example, the neuron signaling is kind of sending the electrical impulse from one neuron to the next neuron, right? Whereas in case of your immune cell, it's about the phagocytosis. Whereas if it is your pancreatic cells, then it will be release of insulin. So the cellular response is different, but the signaling or signal transduction is something which is very common to almost all the cells. Just the cues or the environmental signals that you are recognizing, that you are picking up, and how the cells are responding. So that is the thing that is different and very specific as well. Very specific set of players, very specific set of regulation, very specific set of pathways, and very specific type of cellular responses as well.
Okay, sir. Understood.
Yeah. Okay. So, now let's move to the intermediate filaments, right? So, intermediate filaments, as opposed to the microtubules are more tubular structures. The actin microfilaments are filamentous structures, as we had seen. They are made up of these two protofilaments that are formed as a result of polymerization of actin subunits, which twist to form a right-handed helical actin microfilament. The intermediate filaments are also fibrous structures which are formed as a result of proteins that get supercoiled into higher order assemblies, which we will see. Now, these intermediate filaments are quite unique in several senses as opposed to the microtubules and microfilaments. One of the striking differences is that while tubulin is the main protein which polymerizes to on the microtubules, and actin is the major protein which polymerizes to form the microfilaments. The proteins that polymerize to form the intermediate filaments are quite varied and diverse. For example, it can be keratin, which is found in hair, nail, horns, etc. It can be lamins. It can be several other proteins, right? So that also depends from organism to organism. That also depends from tissue to tissue, organ to organ, as well as within the cell even, that in which cellular compartment, which type of intermediate filament you are having. And interestingly, intermediate filaments are a very unique characteristic of metazoans. Like the earlier organisms do not have the intermediate filaments. And some other unique properties are that while we have seen that the microtubules and microfilaments have a polarity, right? They have one negative end and they have one positive end, and which we had discussed that the negative end and positive end are primarily because of the differences in the rate of polymerization and depolymerization of the subunits that are happening at these two ends. And that also has some biological significance because this polarity allows directional cellular events to happen. For example, directional transport of vesicles or organelles on these cytoskeletal elements, or directional movement of the sister chromatids of the chromosomes during cell division. Intermediate filaments, however, don't participate in any of these directional cellular events, and they do not have this negative and positive polarity. And that also is so because of a reason that the intermediate filaments are actually much less dynamic. The intermediate filaments have much slower filament dynamics as compared to microtubules and actin microfilaments. So what that means is that while you see these continuous trade-off of polymerization versus depolymerization at two different ends, albeit in case of microtubules and microfilaments, in case of intermediate filaments, you don't see that. You see much slower, almost negligible um, dynamics or trade-off between polymerization and depolymerization. That is, once the intermediate filaments are laid at some particular cellular subcellular location, it stays there. And that also has a reason, that also a cellular functional context, that these intermediate filaments are, their major function is to provide the mechanical strength and rigidity that are necessary uh, the for maintaining a particular structure and subcellular localization, right? So that's why they do not change too much. There is a much slower dynamics, and since there is not much dynamics of this polymerization versus repolymerization, they lack a polarity. So they do not have this directional polarity, right? This is an example of these keratin intermediate filaments, which are there in our hair, in our nails, like, right? So there, what happens is that they consist of these alpha helical keratin monomers, right? And then these monomers uh, come together to form a coil dimer, right? And see, then they keep on assembling to form higher order assemblies. Like this dimer then comes together, and two coiled dimers are put together in an antiparallel direction, kind of. Like this is N to C, whereas for the other dimer, it's N to, this is the opposite direction, having N to C. So these two dimers running in opposite directions come together to form the staggered tetramer. Then two tetramers also pack together to form even higher order assembly. And eventually they form this 8 tetramer twisted state, which finally forms the intermediate filament, right? And that which is actually laid in a certain subcellular localization. For example, it may be laid just beneath the nuclear envelope, the nuclear membrane, basically, towards the nucleoplasmic side, which forms the nuclear lamina. So there the intermediate filaments are formed of lamine protein, and those form the intermediate filaments of the nuclear lamina, right? Which is being actually shown here. So here you are seeing that this is the nuclear membrane, right? This is the nuclear membrane. It has two layers, right? And in the nuclear end of this of the nucleus, that is towards the nucleoplasm, just beneath the membrane, you have this elaborate network of intermediate filaments which are made up of lamine proteins, and they form the nuclear lamina, right? Now, one important thing is that which is important to remember is that we have discussed several times that one important function of the cytoskeletal elements, especially the actin microtubules, sorry, the actin microfilaments and the intermediate filaments, is to provide mechanical strength, right? Mechanical rigidity, which is necessary, for example, to maintain the shape of the cell, as well as various different subcellular compartments and organelles, as well as, for example, in the case of the actin filament, it also plays a very important role in changing the shape of the membrane, right? In transducing mechanical force to the membrane, like in terms of causing membrane invagination when the cell is trying to endocyte or something, or in terms of cellular extensions like philopodia when the cell is trying to philopodia or lamellipodia when the cell is trying to move in a certain direction, or for example when the cell is trying to phagocytose something, right? So for that, see, if the membrane and the plasma membrane and the cytoskeleton, right? So if they're separate, if they have no connection, if they're not anchored to one another, then the movement of the cytoskeleton, like the elongation or shortening of the cytoskeleton, will not translate the movement of the plasma membrane, right? So the cytoskeleton, which is lying just beneath the plasma membrane, or for that matter, even this nuclear membrane, right? The intermediate filaments lying, these intermediate filaments made up of lamine proteins lying just beneath the nuclear membrane towards the nucleoplasmic side, or the actin microfilaments, which are located just beneath the plasma membrane, they're anchored also to the membrane via several linker proteins, right? There are several proteins which act as a linker or the anchors between the transmembrane proteins of the plasma membrane and the cytoskeletal elements. Because of that, when there is some change in the cytoskeletal element, there is also a change in the plasma membrane. There can be membrane bending, there can be membrane elongation, there can be formation of this filopodia or lamellipodia, as well as because of that, the cytoskeleton is able to provide the mechanical strength to that membrane which is present at the cell-cell junctions, right? Tight junctions or gap junctions. So that is important. Okay. And also, I mean, in case of the plasma membrane, so that is this picture is also important in this sense that outside the plasma membrane towards the extracellular phase, there is also this extracellular matrix, right? This extracellular matrix is a very interesting thing. The extracellular matrix is composed of proteins as well as polysaccharides, right? Proteins it consists of several glycoproteins, which kind of acts as a glue, right? Which kind of acts as a glue that keeps together the cells, and the extracellular matrix is a very important component of formation of tissues, behind formation of tissues. So it has several proteins, keratin sulfates, heparin sulfates, collagen, chondroitin sulfates, so which are basically glyco proteins. So they are long, there are long polysaccharide chains which are attached to proteins. It can be as well as proteins of the plasma membrane, like transmembrane proteins of the plasma membrane. Okay, so so uh, so we have seen the cytoskeletal elements, right? We have seen the three different cytoskeletal elements: the tubulin microtubules, the actin microfilaments, and the intermediate filaments. Now, among them, we have discussed that the microtubules and the microfilaments are the ones which are polar, which have a kind of a cellular directionality. That is, they have a negative end, they have a positive end. Now, the negative and positive end first, the nomenclature is based on which end is getting more polymerization and which end is getting more depolymerization. But interestingly, as we had also discussed earlier, that in the cell, uh, for example, in case of these microtubules, the negative end is mostly in the microtubule organizing center, which is located nuclearly, that is located just outside the nucleus, right, at the center of the cell. As the positive ends are the ends which are present at the cellular periphery, right? And this directionality of these cytoskeletal elements like microtubules is very important because there are several proteins, right? So cytoskeletal elements are kind of the railway tracks that are present within the cells, but they are very rigid structures as well, right? They do not have too much of motion. They're relatively rigid than others. So there has to be, so they're like kind of the railway tracks or highways. They're kind of those scaffolds on which something else can run. So what is that something else? What are the trains or cars or buses of the cells then, which run on these railway tracks or the roads or the scaffolds basically. So and they they also run in a certain directional way. That is, they run in a particular direction to execute a certain cellular function. So there are several different cellular events, directional cellular events, that is directional movement of membrane enclosed vesicles within the cells, directional movement of membrane enclosed organelles within the cell to ensure a certain spatiotemporal um, uh, kind of organization of organelles within the cells, as well as, for example, during mitosis, you have a directional movement of the sister chromatids, right? They move from the center to the periphery. So there are several different directional cellular events, and now we will try to look into the proteins which are very important in mediating the various different directional cellular events.
Sir,
Yeah, Sean.
sir, can you repeat what is the train or the carts that are moving on the means the cytoskeleton?
I have
Can you,
I have not introduced them. I am going to introduce them.
sir? Okay, sir. Okay, sir.
Yeah. So basically, there are three classes. So these proteins, which are the trains or buses, of the cells that run on these cytoskeletal highways for eventually, executing directional cellular events are very justifiably called motor proteins. Okay. So these motor proteins can be of three, can be primarily of three different categories, of three different types. So one of these motor protein is myosin, and so these are myosin, kinesin, and dynein. Okay. Now to begin with, myosin is a motor protein which primarily runs on actin tracks, right? So it is actin cytoskeleton based motor protein. Whereas kinesin and dynein are microtubule cytoskeleton based motor proteins. Okay. Uh, now, so first we will discuss about the myosin superfamily proteins. So these myosin motor proteins, which are actin cytoskeleton based motor proteins, there are several different myosins in several different myosins in case of human beings. Now there are also cell type specific expression of different myosin proteins, a tissue specific expression of different myosin proteins in human beings. Now, among all these different myosin proteins, for example, you see that the motor domain, the N-terminal motor domain, is mostly conserved, is mostly the same among all these myosin proteins. But this, if you see the structure, we will be able to, like these myosins have two globular motor domains at the end term, then it has these light chains, and then there's a long stalk, right, which actually carries, which either carries the cargo or these stalks help in dimerization or crosstalk between two myosin molecules, allowing movement between the myosin molecules. So there are several different myosin proteins. A motor domain, which is a primary domain that binds ATP, hydrolyzes the ATP, and uses the ATP energy to move, right? This to move on the actin cytoskeletal tracks is the motor protein, is the motor domain of these myosin motor proteins, which are more or less same among the different members of these myosin superfamily proteins. However, the stalk domain is quite different among the different myosin proteins, probably because of the different functions. So that gives them different functional capabilities, which also and because of that, different tissues and different cells which have different cellular needs or cellular functions to be executed express different myosin proteins as suitable. So myosin proteins have this, as we already have seen, has two N-terminal globular. So in all of these motor proteins, you will see that there are some head domains, which are basically the motor domains. So these are globular head domains or motor domains of the myosin protein, which bind the ATP, which bind ATP, and in their ATP bound state, they bind to the myosin cytoskeleton. But then what happens is there is an ATP hydrolysis into ADP and PI, and that causes a conformational change in the myosin proteins globular domain. And because of this change in conformation, what happens is it gives a power stroke. It literally, it's like a human being moving. When it hydrolyzes one ATP, its feet moves as if for a certain distance, right? There is a conformational change which allows it to move a certain distance. And so that's the function. That's a major function of this N-terminal globular head domain of the myosin proteins. Whereas, and there is this light chain which connects the coiled coil region to the globular head domain. And this long coiled coil region consisting of two alpha helices, which forms two alpha helices, which are basically two heavy chains. This long coiled coil domain is actually the stalk. Like you can think that it has two globular domains, and on top of the globular domain, it has a long stalk. And on this long stalk, there can be this vesicle, right? This membrane enclosed vesicle, which will be the cargo that it may be transporting, right? It may be moving the actin cytoskeleton, or this stalk we will see in the next slides, right? For example, in case of myosin 5, you can see that you have this. Actin cytoskeleton tract, which is the highway, right? and on that your myosin 5 is there. So myosin 5 is binding. Myosin 5 is binding. It has its globular motor domains which will bind with ATP. Upon binding with ATP, it will bind to this actin cytoskeletal element, and then what will happen is there will be ATP hydrolysis into ADP plus PI, which will release energy and also cause a conformational change. So because of this conformational change, there will be a stroke. It will move a certain distance, and on top of this stalk, you can see the cargo binding stalk or the tail basically of myosin. You are seeing that a membrane enclosed cargo is bound. And so basically, in simple words, upon hydrolysis of one molecule of ATP, this cargo will move one step. Like one step will be some nanometers, right? 5 nanometer or some nanometer. That also depends from myosin to myosin in this direction. So there also comes this directionality for example, from the negative end to the positive end over the actin cytoskeletal element by this myosin. So the directionality will also depend from myosin to myosin. Yeah. And the other thing that myosin performs, which is an important function of the myosin, is that which you see in case of which you see in two important cases within the cells. One is the skeletal muscles, right? Where myosins, this actin and myosin are very important components for the muscle contractility and relaxation. That is for muscle contraction and relaxation, without which we would have not been able to even move and do any of our normal activities, right? So there what happens is you have in the skeletal muscles, you have the actin cytoskeletal elements, right? And you have the myosins, which are this is a myosin 2. So here you can see that there are several myosins here, right? Like these are the two globular heads of one myosin molecule. And here you see its long this tail, or this basically coiled tail. Here you see one more myosin molecule, which is again bound to the other actin cytoskeletal elements. There are two actin cytoskeletal elements. There are myosins which are bound to both the actin cytoskeletal elements. And in between these coil regions are allowing the myosins, right? The myosins to dimerize again, to interact with one another. And when there will be so they are now ATP bound, but when there will be ATP hydrolysis, what they will do is that they will give a stroke, like this, which will cause the muscle to, for example, during muscle contraction, it will cause this actin to move in this direction, and it will cause this actin to move in this direction. So basically, there will be muscle contraction, right? Whereas in case of muscle relaxation, what will happen is that this one power stroke will cause the actin to go in this direction, and one power stroke of this myosin will cause the actin to go in this direction, causing muscle relaxation, right? Both of which are necessary for our normal physiological functions. So that is how myosins also play not only in the cargo transport on the actin cytoskeletal network, which is located just below the plasma membrane, but also myosins play an important role in muscle contractility, right? Muscle movement, muscle contraction, and relaxation. Not only this, myosin is also very important in this activity of the actin cytoskeletal network, which is um, which is basically modulating the shape of the plasma membrane, or conveying the force that is generated as a result of cytoskeletal reorganization to the plasma membrane. For example, the movement of these myosins, which are connecting various different actin cytoskeletal networks, is necessary for the change of the plasma membrane. For example, the bending of the plasma membrane, as you can see in case of this endocytosis event, the invagination of the plasma membrane, or in case of cellular morphology, in case of cellular motility and movement, but there is the formation of filopodia or lamellipodia. There also this coordinated function of both actin as well as myosin is necessary for conveying the force that is generated as a result of actin reorganization to the plasma membrane uh, to cause the change of shape or bending of the plasma membrane. for cellular activities. As well as we have seen that this cargo movement, in cargo movement also myosin plays an important role on the actin cytoskeletal, like actin microfilament tracks.
Sir,
Yeah, so
question please. Yes, sir. From where the myosin get the ATP?
myosin gets the ATP from the cytoplasm.
Okay, sir.
Yeah. So, yeah, now just as we were mentioning while discussing about the different myosin members of the different myosin proteins of the myosin superfamily, that you can see one interesting thing that like we said that the N-terminal motor domain, right, which is the ATP binding domain and which will eventually bind the actin cytoskeletal element, is more or less the same for all the members of the myosin proteins, which should be also because I mean this is a conserved function, right? Every myosin has to bind this actin cytoskeletal elements in the presence of ATP. So that domain is quite similar across all these similar kind of, you can say, evolutionarily conserved among all these myosin members, different myosin members of the myosin superfamily. But the N terminal, this C terminal tails, basically those heavy chains, those coil regions which form the stalk or the tail of the myosin, is different among the different myosin members. So which can also be understood in this context that these different sequences can give them different capabilities. For example, some binds the cargo, whereas some of these tails allow the interaction between two different myosin molecules, which is necessary for the muscle movement, that is muscle contractility or relaxation, or can be important in the transduction of the mechanical force to the plasma membrane that we saw. But interestingly, there is also one interesting thing in this, that how. So, for example, in case of muscle movement, that is muscle contractility or relaxation, there you can see that the length of the tail, right? You can see that there are different lengths of the tail. The length of the tail, this is myosin 2, this is myosin 5. In myosin 2, it has a shorter tail, whereas in myosin 5, it has a relatively longer tail. You can see that since it's a shorter tail, the one power stroke creates a much smaller displacement, right? It creates the one power stroke with the shorter tail of myosin 2 creates a displacement of around 5 to 10 nanometer. Whereas for myosin 5, which has a relatively longer tail, one power stroke or one swing of the head actually causes a bit more or longer uh, kind of traversing of length, uh, which is around 30 to 40 nanometers. So the lengths of the tails of these myosin proteins, which are different among the different myosin members, um, also becomes important in this aspect, that in a single power stroke, how much of length you are traversing, right? That is how many power strokes, which is in other word, gives you this idea that how many power strokes will be needed to achieve a certain to achieve the traversing of a certain length that is necessary for the eventual muscle movement and eventually execution of some activity. So that's why there are different tissues having different muscle expressing different members of the myosin superfamily depending on the function that is to be executed. Next up, so after we have seen the motor proteins which are dependent on the actin cytoskeletal elements, that is actin microfilaments, now we are heading to the class of motor proteins which are dependent on the microtubule cytoskeletal elements, right? So the cytoskeleton, microtubule cytoskeleton-based motor proteins primarily consist of the kinesins and the dyneins, right? So kinesins, now structurally, these kinesins and dyneins are also quite similar to that of myosin. So here also what happens is in this image, if you can see, then we will also see in the other schematics in the upcoming slides that kinesin also has two N-terminal globular motor domains, right, which bind ATP, and they will uh, then they bind these microtubule cytoskeletal tracks, and on the other, then they have this tail, right, on the end of which it has the cargo binding domain. Okay, so in the N-terminal globular heads or the motor domains will bind the microtubules in the presence of ATP, whereas and there will be long tail which is flexible, which allows the cargo to move, wobble a bit while it is moving on the microtubule track, for facilitating the movement of the cargo within a very crowded cellular environment. And then in the C-terminal, it has the cargo binding domain. But this picture also very nicely shows that how big membrane enclosed vesicles do these motor proteins carry um, on these microtubule tracks, right? So you can see that the motor proteins can carry very relatively much larger cargoes like membrane bound vesicles, which are emanating from the Golgi, which has to be transported to the plasma membrane, or vesicles which are formed as a result of endocytosis from the plasma membrane, which has to be transported to the Golgi or to the lysosome, or the cargoes can also be membrane-bound organelles, right? And however, do consider this fact that it's not the case that a single cargo has just a single motor protein. Several motor proteins can be associated with a single cargo. And several motor proteins of different kinds can also be associated with one particular cargo, like both the kinesins and dyneins can be associated. But the number of motor proteins eventually decide that which direction they're going to move. So if you look at the structure, so as I already mentioned, that this N-terminal, you have these globular motor domains, which binds the ATP and binds the microtubules and skeletal elements. And then there is a long stalk or tail, um, and eventually in the C-terminal, you have this cargo binding domain, right?
Sir,
Here, yeah, Shom.
sir, sir. Basically, what are you telling about the cargo binding? What is the cargo? What are you saying that about?
Cargo, basically, so cargo can be quite diverse. For example, um, cargo mostly for these kinesin proteins are membrane-bound uh, subcellular compartments, right? So in cells, for example, if you look at the protein secretory pathway or the protein sorting in the protein secretory pathway, or for example, various other pathways as well. Like in cells, what happens is the proteins are secreted, the proteins are synthesized. Yeah, so the proteins are synthesized within the endoplasmic reticulum, right? The ribosomes synthesize, I mean, the ribosomes are present in the cytosol, but the proteins which are meant to be secreted out, so they are kind of co-translationally delivered into the endoplasmic reticulum by the ribosomes. Then these proteins go to the Golgi, and from the Golgi, what happens is, from the trans-Golgi network, these proteins, which are finally to be sent to the plasma membrane, for example, right? The Golgi is present close to the nucleus, right? But from the Golgi, there are several proteins, which are plasma membrane proteins, which have to be transported to the plasma membrane, for example. So how do they get there, right? So what happens essentially is that, um, for example, this is your cell, this is your this is your cell, this is your nucleus, okay? This is your endoplasmic reticulum, for example. And then you have stacks of Golgi, right? And these are all membrane enclosed subcellular compartments, right? Now within the Golgi, you have one particular protein, which is a plasma membrane protein, which needs to be delivered to the plasma membrane. So how do you deliver that? So essentially what happens is that you have these microtubule cytoskeletal tracks, right? Which are distributed all over the cell. And this is the plus end, this is the minus end. So from the Golgi, what happens is several small vesicles, right? From the Golgi, several small vesicles are formed within which, for example, your plasma membrane protein is present. Okay. And then what happens is then these kinesin motor proteins are there, which bind to certain specific proteins, transmembrane proteins of this particular vesicle. Okay, which is having the plasma membrane protein that is to be delivered. That was formed from the Golgi and needs to be delivered to the plasma membrane. So then this motor protein kinesin moves in a particular direction. It only moves in the plus direction of the microtubule. So it actually carries this vesicle on its head, in this case on its tail. So it carries this protein on its tail and carries this vesicle actively using ATP energy from the negative direction to the positive direction. This is the positive direction of the cell to the periphery of the cell, where finally this vesicle fuses with the plasma membrane and the plasma membrane protein is eventually delivered to the plasma membrane. So primarily, that's why I was saying that cargo is what? Cargo is basically, for example, if you have a truck, then whatever you load into the truck is your cargo, right? So for the motor proteins also, motor protein is like a truck, right? So whatever you load on the motor protein is its cargo. But typically the cargo for these motor proteins are membrane enclosed subcellular compartments. So they can be membrane enclosed, they can be these membrane enclosed vesicles which are being formed from the Golgi. They can be subcellular organelles like the lysosomes or mitochondria, which also need to be transported in a certain direction or anchored to the microtubules to maintain a certain spatial localization of those organelles within the cell, right? Like even the Golgi or the endoplasmic reticulum, why are they present why are they present close to the nucleus and not close to the periphery? Because they are also actively kept there. They are not present there by default. They're actively kept there by these several of these motor proteins or several other proteins which are anchoring them to the cytoskeletal elements, right? And how is this specific movement of these membrane enclosed organelles or membrane enclosed vesicles happening within the cells? For example, that is also executed actively using this ATP hydrolysis energy by these motor proteins, right?
Okay, sir. Understood.
Okay. Yeah, so basically this cargo binding domain in the C-terminal part of these kinesins or even in case of the dyneins, so they essentially will bind to some transmembrane protein, right? Some domain of one or more transmembrane proteins which are present on the surface of the cargo, right? That helps in recognition of the cargo, which for example is the membrane enclosed organelle or membrane enclosed vesicle by this kinesin protein, which will then carry it by moving on the microtubule tracks in a certain direction. So as of now, the kinesins that we are discussing, the kinesin causes anterograde movement. That's what I mentioned, that kinesin causes anterograde movement of these cargos. That means it moves cargos to the positive end of the microtubule. Okay, the positive end of the microtubules is present towards the cellular periphery. That is, kinesins are anterograde motor proteins which move cargo to the plus end of the microtubule. That is from the center towards the periphery. It moves cargos towards the periphery. Okay. Right. Yeah, and interestingly, I mean, this is also an important point to ponder that these motor proteins, they utilize they utilize the ATP energy, right, to actively move cargos. So this motor domains, right, this motor domain is a domain which binds ATP as well as it has the capability to hydrolyze ATP, right, um, to generate the energy that is necessary for the conformational change, which will allow again this domain to move forward for allowing the movement of the um, motor protein on the microtubule tracks. But the cell has to regulate, the cell has to ensure that this motor protein, for example, the motor domain of these motor proteins, be it kinesin or dynein, that they are not hydrolyzing ATP randomly, or they are not wasting ATP. What I mean to say is that they are not hydrolyzing, they are not binding and hydrolyzing ATP when the cargo is not bound on the motor protein. So there is a regulation, auto-regulation or self-regulation in these in case of these kinesin motor proteins. So what happens is that when there is no cargo that is bound to the motor protein, the motor domain is inactive. That is, it does not have the capability to hydrolyze the ATP. But when the cargo binds to the cargo, so there is this interaction between the N-terminal motor domain and the C-terminal cargo binding site, which causes the protein to fold. You know, the two hands of a single protein which are tied to one another. So none of the hand is able to do the function that it is supposed to do. But when the cargo is present, the cargo binds to the cargo binding site, right? The protein, for example, the protein which is emanating out of this membrane bound vesicle now, which is the cargo, which is binds to this cargo binding site. Because of that, what happens is there is a conformational change of this entire protein. So this self-regulation, this N-terminal motor domain then actually gets freed from the C-terminal cargo binding site, and then the N-terminal motor domain becomes active. That is, it is now capable of hydrolyzing ATP, as well as then what happens is that these two kinesin molecules, they dimerize. There are two N-terminal motor domain heads, which in the ATP bound form now bind to the microtubule cytoskeletal tracks, and then they are now, since they're active, they hydrolyze the ATP to get the ATP energy, which now helps them in moving forward in driving the cargo on the microtubule tracks in a certain direction. Ravin is asking something. Yeah, Ravin, your exam will be in MCQ mode only. Okay, so so this is just a comparative view of these two motor proteins, myosin and kinesin. So as we have seen that myosin is an actin cytoskeletal element dependent motor protein, whereas kinesin is a microtubule cytoskeletal element dependent motor protein. Myosins, there are several different categories, several different uh, types of myosin as well as kinesin. So depending on that, their functions, they have slightly different
So
functionalities.
I will, and then
Myosin,
I will call you back.
yeah. So there are different members of myosin superfamily as well as kinesin superfamily, and depending on their subtle differences in the protein structures as well as protein sequences, they have subtle differences and functionalities as well. So myosin plays important roles in muscle movement, muscle contractility and relaxation, in transduction of the force that is generated as a result of cytoskeletal reorganization to the plasma membrane, thereby contributing and regulating the shape of the plasma membrane, as well as cellular movement and motility, like formation of these lamellipodia and filopodia, um, as well as myosin plays an important role in cargo transport on the actin cytoskeletal network, right? Whereas kinesin primarily is an anterograde microtubule cytoskeleton based motor protein. That is, it is necessary for movement of cargo. There can be diverse cargoes, but primarily these are membrane enclosed vesicles. It can be membrane enclosed vesicles. It can be Golgi vesicles. It can be other membrane-bound organelles, which are moved by the kinesin motors in an ATP energy-dependent manner in the plus end direction of the microtubules, that is towards the periphery of the cell, right? Now we will be heading to the other class of microtubule cytoskeletal element based motor protein, which is the dynein motor protein, right? So dynein, as opposed to the kinesin motor protein, actually drives the minus end directed movement of membrane enclosed cargo, membrane enclosed vesicles, membrane enclosed cargo. That is, they drive the movement of this membrane enclosed cargo in a retrograde manner towards the minus end of the cytoskeletal elements, that is from the plasma membrane towards the nuclear side of the cell, right? That is the movement that is being actively driven by the dynein machinery, dynein motor machinery, right? Again, actively, because dyneins also need the ATP energy. Structurally and domain-wise, they're also quite similar to the kinesin. However, dyneins need one accessory protein complex, which is called the dynactin protein complex. And the functional dynein is actually the dynein-dynactin protein complex, which binds ATP, which binds cargo, and the N-terminal head groups will be bound to the microtubules, and then they will catalyze ATP hydrolysis, use the energy that is released as a result of ATP hydrolysis to drive the movement of cargo in the negative end, towards the negative end, that is towards the nuclear end of the cell, towards the negative end of the microtubule, which is located perinuclearly, right? That is towards the nuclear end of the cell. So domain-wise, just like the kinesin and myosin, dyneins also have one N-terminal head domain, which will have this microtubule binding domain, as well as the ATP binding domain, right? Which will also hydrolyze ATP to release the energy for driving the active transport. And it also has the cargo binding region, which will bind the specific proteins, the transmembrane proteins that are emanating out of the membrane enclosed cargos, right? There are some chat box messages. Yeah, somebody is concerned that her name is not being reflected in the attendance sheet. I mean, I conduct the sessions on Zoom, so I don't upload any link where the you know, who has attended is being shown. So I just mentioned the number of participants. You don't worry. I mean, this is not of any concern. It's fine. Okay, now dyneins actually, apart from this very important function of retrograde transport of cargoes within cells, that is movement of cargoes from the plasma membrane towards the nucleus, right? Towards the nucleus of the cells. Um, mm, Dyneins have one very one important specific one important quite specific function. For example, in the flagellar movement of sperm, which is necessary for propelling the sperms and is important for fertilization. So the sperm has a flagella which has a very unique arrangement of microtubules. Okay, there are these microtubule doublets which are present at the periphery, and there is a central microtubule doublet. Now, dyneins are present between these microtubule doublets, right? In this is the inner dynein arm. You can see this inner dynein arm, which exists between the outer dynein and in this inner central singlet microtubule. So this dyneins actually um, move these two microtubules. Okay, this two moves the microtubules in a different in a different directions, that causes a waving movement, right? That causes a waving movement on the spawn flagella, and necessary for the strokes, the power strokes of the spawn flagella, which allows the spawn flagella to the sperm to propel in a certain direction towards the egg or the ovum, and is very important for the fertilization of the egg by the sperm. Yeah, so that almost concludes our discussion on the important motor proteins, myosin, kinesin, and dynein. But one interesting uh, last uh, thought to ponder is that the cells, most of these like which kind of which kind of motor proteins are better, or what are the specific sets of advantages versus disadvantages that come with the motor proteins that have unidirectionality, for example, kinesins and dyneins, or bidirectionality, which is more prevalent in case of myosin. Okay, so the thing is that it is not that one particular like whether it is better or not depends on what purpose the motor protein has to execute, right? Some of these motor proteins, especially in case of myosin, the bidirectionality is more helpful, whereas in case of this kinesin and dynein, which need to move cargoes in a very specific manner, either towards the plus end, that is towards the periphery of the cell, or towards the minus end, um, that is towards the nuclear section of the cell. So these kinesins and dyneins, for them, unidirectionality works best, whereas in case of myosins, maybe the bidirectionality is more suited for a bidirectional movement um, of on the acting cytoskeletal filaments, as well as, however, bidirectionality, also bidirectionality of a motor protein, like unidirectionality in case of myosin uh, would have created one set of challenges, and for example, bidirectionality in case of kinesin and dynesin would have created other set of challenges. They would have become more error prone, more set of regulatory proteins would have been necessary for properly or stringently guiding and directing these motor proteins. In case of kinesin and dynesin, if that would have been the case. So whether cell has preferred unidirectionality, unidirectional motor proteins or bidirectional motor proteins is stringently dependent on the function of the overall function of a particular tissue type or cell type um, that is concerned. Okay, so this brings us to the end of today's session. So if you have any question, I will be happy to answer. I would like to mention discuss about one question which was there in the discussion forum. So somebody had asked that whether the plasma membrane is selectively permeable or semi-permeable. And the second question was that the plasma membrane is actually made up of a lipid bilayer, right? And there is this hydrophobic hydrocarbon chains which are present in between, like inside the lipid bilayer. So how do water molecules, which are polar molecules, that is, they will not be allowed by this hydrophobic core to come in. So how do they traverse the lipid bilayer? The second question is very important. So the plasma membrane is actually selectively permeable because it has a set of proteins. It has a set of transmembrane or integral membrane proteins, as well as a set of different transporters like ATP binding cassette transporters and other proteins, which selectively allow some molecules to get in or go out. That is, they selectively allow the movement of certain molecules and also in a particular direction. That is, whether from the extracellular space to the cytosol or from the cytosol to the extracellular space. That's why the plasma membrane should be considered selectively permeable. Semi-permeable membranes are like membranes which depending on the chemical properties of the membranes, as well as allow certain molecules to pass, whether does not allow other molecules to pass depending on the concentration reading. So plasma membranes are selectively permeable because they have a dedicated set of proteins that allow the movement of only particular set of specific molecules, and that too in a specific direction. From the extracellular space to the cytosol, or from the cytosol to the extracellular space. And the second question is, the answer to the second question, that how does water move in or go out during osmosis, in case of the cells, through the plasma membrane? So the answer is that there is a dedicated set of proteins which are called the aquaporins. Okay? There is a dedicated set of proteins called aquaporins, which acts as a channel protein. It's a integral transmembrane protein or integral membrane protein, which forms a channel or pore within the membrane, which specifically allows the movement of water depending on the osmotic pressure, basically inside in the cytosol and the extracellular space. So that allows the movement of water through the plasma membrane. Otherwise, the plasma membrane being a lipid bilayer, which has a hydrophobic environment inside the lipid bilayer, does not allow the movement of water. Water can only move through this transmembrane or integral membrane channel protein, which is the aquaporin in case of, and the direction depends on the osmotic pressure in the extracellular space versus the cytosol. Okay, I hope you were able to understand the concepts and the topics of week 6 of this NPTEL course, Introduction to Cell Biology, and hope that you will be able to do the assignments as well for this week. So if you don't have any further questions, then let's end for today. Hope to see you all in the next session.
Can you say once retrograde?
Retrograde? Yeah, sure. Basically, retrograde movement is movement in the movement in the negative towards the negative end of the microtubule. So retrograde movement is mediated by the dynein-dynactin complex. So primarily the dyneins are the motor proteins which move the membrane enclosed cargo in the negative, towards the negative end of the microtubule, opposite direction of that of kinesin. And since in the cell, the negative end is where? Negative end of the microtubules are close to the nucleus. The dyneins basically move cargoes from the plasma membrane towards the nucleus. For example, when the cell is undergoing endocytosis, right? For example, the cell will engulf something, endocytosis or phagocytosis. Then what happens is, after it engulfs, what happens is there is formation of this vesicle, okay, within which your engulfed thing is present. For example, bacteria or something else. Then that has to be moved towards this, moved by the dyneins towards the nucleus. Now, most of the acidic lysosomes, which are also degradative lysosomes, they are present in this perinuclear region. Okay. So there, then what happens is this cargo fuses with the lysosomes, and within the lysosomes, your bacteria is degraded. Or this retrograde movement may also be important, for example, in recycling of receptor proteins, which were present on the plasma membrane, to the trans Golgi. Now, your Golgi is present near the nucleus, and your receptor is present on the plasma membrane, and you need to recycle this receptor back to the trans Golgi, for example. Then what happens is, there is invagination of this membrane. A vesicle is formed within which this receptor is now present, and then you have these microtubule tracts, which are the negative end in this direction, and dyneins carry these vesicles in this direction to the trans Golgi, and thereby then there is membrane fusion between the vesicle and the trans Golgi. So eventually there is a delivery of this receptor from the plasma membrane to the trans Golgi or to any other compartment, basically, which is located perinuclearly, that is around the nucleus, because dyneins drive the retrograde or the negative, or the movement towards the negative end of the minus end of the microtubules. Is it
Okay,
clear?
sir. Clear. Yes, sir. Clear.
Okay. Okay. Bye then. See you in next class.
Thank you, sir.